My name is Dr. Tiffany Bellomo, and I am in vascular surgery at the Massachusetts General Hospital, but I'm working on a lot of different projects to look at the genetics of SCAD, and I have no disclosures. So today I'm going to talk first about the heritability of SCAD, then how we are understanding this through genetics, what we don't understand, and what we can do about that. And so first about the heritability of SCAD, I wanted to take a step back, and I know that Vonda gave a great overview of her co-diagnosis of FMD, but just looking all the way back at genes and how they impact SCAD, we know that DNA is found in every cell of your body, and it contains these genes, which are like instructions. And this DNA on screen left, you can see, will tell your genes how to make proteins. So these proteins actually do most of the work in your body, and some of them help build your arteries. And what are arteries? Well, they are made up of collagen, elastin, other proteins. And when the genes aren't giving the right instructions to make these arteries, the body can make proteins that don't work properly. And that's something that was investigated in this paper below. So taking a step forward and saying, well, hey, if we have these genes, can we identify which genes are running in families? So the heritability. And in this study on the screen left, you can see, there's a lot of information here, but it's pedigrees, which draws who has SCAD in each individual family. And there were 132 families that were followed. And the people who were originally diagnosed with SCAD are represented in black. And the ones that had a gene that corresponded to SCAD, or a known gene, are outlined in red. And you can see that's not very many out of all these families. So only 4% had some sort of rare genetic variant. And so what we do know is this green overlap with SCAD on the bottom left, where there are rare genetically triggered arterial diseases. And we also know what we just heard that Vonda had was FMD, which is also an overlapping trigger of SCAD, which kind of goes with the two-hit hypothesis. But we want to know more about what else could contribute to the two-hit hypothesis. What other variants can we discover that cause SCAD? Because I just talked about what was known, but what about what we don't know? And so that's where the advances in studies have begun. So there's a lot of different research on the genes, genetics of SCAD that have been performed, one of which I want to highlight here, which is a GWAS. So, well, what is a GWAS anyway? So to explain this, I'm going to take you through 4 individuals who have DNA, as we previously talked about, and I have their strands of DNA outlined here. And what is a SNP? Well, these are a column of letters that represent a single letter in a strand of DNA. And we can look at these individuals and notice that individual 2 in this specific place has an A, where others have a G. So at that time, we can ask, hey, well, does having the letter A increase the risk for disease compared to letter G? And we can do that and ask that same question for every single spot or every single nucleotide polymorphism in the genome. And when we ask that question and run that test for every single spot, we come up with something called a Manhattan plot, which this is from the latest study, GWAS of SCAD. And what's a Manhattan plot? Well, you can see on the x-axis, we have chromosome. So imagine a bunch of those lines of DNA all lined up representing all of your chromosomes. And on the y-axis, we have how significant is it? And what we see is in this analysis of over 1,900 cases and 9,000 controls, we identified 6 specific positions or changes in the genome that represent that change in letter that were associated with SCAD. And when we look at those individual changes, we then have to ask the question, well, hey, out of everybody in a registry with SCAD, does that change result in a change in a protein? So we asked that question with respect to one gene, and we looked at fibrillin collagen, which again is part of what makes up your arteries. And we looked at that fibrillin collagen gene, and we looked in animals and found that these collagen defects do cause SCAD. So that's how we take these genetic corollaries to the lab, to animals, and understand, does this actually cause SCAD? And so with all this knowledge and technology, what do we not know? Well, some of the limitations of each study are that they have less than 2,000 participants, and almost all of them represent Europeans. So that's one ancestry group. And we've determined that we only know 24% of the genetic contributions to SCAD. So that leaves another 75% unknown. And discovering these genes will help us understand not only the causality that I showed previously, but also will help us develop new therapies. And so, given all these gaps that remain, what can we do in the future? Well, new technologies will help us with discovery, including expanded genetic analysis, looking at risk prediction, and therapeutic development that I just discussed. But to do that, we need to partner together to get more people in a registry. And so what we've done is there's a study called GVASC that looks at the genetics of vascular disease. And we worked closely with SCAD Alliance and multiple iSCAD investigators that you'll see here today. And what we looked at was, hey, can we partner together to get genes and understand which genes are causing SCAD? And how do we do that? Well, GVASC is a study where participants will provide saliva here on the left, and we will take the saliva and extract DNA from it. We will compare the DNA of people with SCAD and without SCAD, and then identify those differences that I talked about before. And hopefully together that will help us identify more causal genes to identify what else is contributing to the two-hit hypothesis that was previously discussed. And when we identify more genes, we can do more wet lab experiments. So there have been a lot of advances with regard to these experiments, including things called cell painting and flamingo assays, some of which you can see on screen left, where the blue represents cells. And we're able to see with our eyes what's going on with the proteins in these cells. There's also a very cool experiment that's going on now where we are able to take a mouse heart and look at the coronary arteries of that mouse heart. And what do we do with that mouse? Well, we are able to go into the genes of that mouse, make that change that we found in the GWAS, one of those 16 different letters, and then see how that impacts the coronary arteries of a mouse heart so we can understand how that works. And so just to review, the current challenges included limited sample diversity, where we're looking to expand our collaboration as the solution, some mechanistic gaps where we're looking to include cell, animal, and imaging models, the genetic complexity of rare and common variants, where we're trying to use our now integrative discovery approaches. And all of that is to say that it's not without pause that we want to thank you guys and the patient participants in these studies to help us learn how we can understand genes better and how we can use that to really create therapies and help you guys. So thank you so much for allowing us to participate in this webinar.